Dr. Gallo did his bachelor in molecular biology and biochemistry at Simon Fraser University under the supervision of David Bailey. He did his PhD in medical genetics at University of British Columbia under the supervision of Don Riddle's lab. And he did his postdoctoral fellowship at Six Kids in Toronto under the supervision of Dr. Peter Dirks. And he's currently he's been an assistant professor at the University of Calgary since December in 2015. So welcome, Marco, and looking forward to your talk. And you're muted if you didn't know that.
Okay, yeah, I was actually testing. Perfect. You guys are paying attention, that's good. So I'm glad that, Anna gave her talk before me, because I think the kind of data we're presenting today kind of speak to the two different souls of our lab. Okay, so the more genomics, more bioinformatics side of the lab, and then the talk I'm going to give is going to be more about how we are applying these genomics tools we have to understand the biology of GBM. Okay, and also I really want to emphasize that the story I'm presenting now is also was also driven by Anna. Okay, and so today's really the Anna show in a way. Okay, so as Anna was mentioning in her talk, the focus of our lab is really on both pediatric and adult brain cancers, with a special focus on GBM. And GBM is an incredibly heterogeneous disease, also intratumorally. And so the way we like thinking about this cancer is as in the context of a heterogeneity matrix. Okay, so there are different levels of heterogeneity that really come together in this tumor type. And one classical example of tumor heterogeneity is on the genetic side. Okay, so there are on the genetic level. So there are obviously each tumor is a collection of multiple genetic subclones that coexist in a relatively similar environment in each tumor. But then what we've been reporting over the years, and other people have as well, is that there is also a certain level of epigenetic heterogeneity in these tumors. Okay, so there are multiple layers of heterogeneity at the molecular level. And I'm just providing here some examples of epigenetic heterogeneity. So we observed this kind of heterogeneity both at the levels of DNA methylation, at the level of histone post-translation modification. So here you see, for instance, cells that are highly positive for K4 methylation, others that have lower levels or undetectable levels. And it also extends all the way to histone variants. So here is as an immunofluorescence on a tumor specimen. So it's a primary tumor resection for total histone 3. And as you would expect, every cell has got histone 3, virtually all of them. But if you look at the specific histone 3 variant called H3.3, you see that only a subpopulation of cells have detectable levels of this protein. Okay, so there is a high degree of heterogeneity. What we have been thinking over the years also is that this epigenetic heterogeneity we're seeing is not noise, but it's probably meaningful, right? And how we think about this is, there is a very well-established, strong link association between epigenetics and cell function. Okay, and that's why your epidermis is different than your blood cells, right? Because the epigenome is at play. And so we've been thinking of the idea that the epigenetic differences we're seeing inside a tumor really reflects some kind of epigenetic hierarchy. Okay. And this feeds onto a stemless cancer stem cell hierarchy that we've been studying for over a decade. And the idea here is that there are probably some chromatin and epigenetic states that are conducive to cells being programmed to be more stem-like, have some kind of stemless properties. Key among them, self-renewal and tumor initiation. Okay. So, whereas other cells in the tumor might not have these specific chromatin states, and therefore they cannot self-renew and propagate the tumor, both in vitro and in vivo. Okay. So the problem my lab is really focused on is this problem of stemness. I think it's recognized in many different tumor types, but it's definitely a major issue in the context of GBM. And my former postdoc supervisor, Peter Dirks, really was the first one to purify the cells that were stem-like back in 2004. And again, I want to really emphasize that, cancer stem cells are dangerous on two different aspects. Number one, self-renewal is indispensable for the long-term tumor growth repopulation potential, especially recurrence. And that's definitely a problem in GBM. Recurrences almost universally kill the patients. Once a tumor recurs, there's nothing you can do clinically, or very little you can do clinically. And part of the reason is stem-like cells tend to be therapy resistant. And there's therapy resistant in different ways. So they actually have this amazing ability of inducing pathways that help them respond to chemotherapies. They upregulate DNA damage pathways that make them less sensitive to radiation therapy, but they also have some inherent properties that make them naturally therapy resistant. And one of them is the fact that they're relatively quiescent or slow cycling. Okay, and this was something that, again, Peter Dirks published a beautiful paper on in Nature in 2017, really showing this population of cells being slow cycling. So you can imagine that most of the treatments that the cancer patients receive are really predicated on the high proliferation rates of cells in the tumor. If a cell is slow cycling, the damage you can induce via chemotherapy or radiation therapy is not going to inflict as much damage on a slow cycling population. Okay, so these cells are really, really a clinical problem and a biological problem. And the good news is we know epigenetics is involved in making them what they are. And so in a way, it's a tractable problem. We just need to figure out how all the pieces come together to make them stem-like. At the same time, though, as everybody knows, targeting nuclear proteins is a major problem, and it's really difficult to bring a drug that targets a prescription factor, let's say, to the clinic. There are examples, obviously, of this happening, but it's not an easy task. So I also wanted to give you really an idea of, practically speaking, what this means. Okay, so there's over 10 years of literature on cancer stem cells, but I really like this piece of data, these two panels, because they really emphasize how cancer stem cells are really different from the non-stem-like cells from a tumor. So here it's data from Brad Bernstein lab in Boston, and you can see you can inject as low as 50 cells that are cancer stem-like cells from a GBM, and the cells engraft, the tumors grow, they kill the mouse. Okay, and then you can inject 100,000 cells that are non-stem-like, and the mice don't die. Okay, but if you take these totally differentiated non-stem cells and you reprogram them by adding 4 transcription factors in a very Yamanaka-like experiment, all of a sudden the cells become tumorigenic again, they self-renew, and they kill the mice. Okay, so epigenetics plays a big role in the biology of GBM stem cells. And the particular aspect of epigenomics and chromatin biology that my lab is really interested in are histone variants. Okay, so I think histone variants and histones in general, I think they've always been important, but it became a little bit more sexy when a member of the network, Nada Jabado, published this beautiful paper in Nature in 2012 showing stereotypical mutations in histone 3.3 at 2 key residues of this protein. These mutations in histone 3.3, in this variant of histone 3, occur almost exclusively in young kids with high-grade gliomas. And are incredibly rare in adults. They occur in less than 1% of adults. Really predispose the tumor to becoming aggressive. Okay. So mutations are rare in adults, but what we showed a few years later was that histone 3.3 plays a role in the stemness hierarchy in adult GBM. Okay, so what we really found was that histone 3.3 levels were very low in the stem-like cells. And then if you increase the levels of histone 3.3, or if you look at differentiated cells, so histone 3.3 was basically antagonizing stemness properties in GBM. Okay, so this kind of firmed into our mind the idea that histone variants actually play a role in the biology of these awful brain cancers. And we started thinking, when I opened my lab, I started thinking about the idea that just like there is a histone code in normal cells, there might be a histone variant code in GBM. Okay, so what I mean with this is that there might be specific histone variants that are promoting stemness properties in stem-like cells, and other histone variants like histone 3.3 that antagonize stemness and are more present in the differentiated cells. Okay. So long story short, basically, we started working on one other histone variant that we think has got a strong role in the biology of adult GBM. And this is a specific variant of core histone H2A, which is called macro H2A2. Okay, so this is the only slide I'm going to have about the background for H2A. There are two genes encoding macro H2A variants. These are H2A-FY and H2A-FY2. I'm not going to go into the specifics, but these encode proteins that have histone domain, and they also have a longer domain which is called the macro domain. Okay, so imagine what happens to chromatin when you exchange core histone H2A, which is about 17 kilodaltons, for one of its variants, which is 40 kilodaltons, and there's this big macro domain sticking out of the nucleosome. Multiple papers have shown that this kind of substitution with the histone variant really changes chromatin structure and the transcription levels of downstream genes. Okay, so macro H2A2, H2A encoded by H2A-FY2. Okay, so this is what we really need to remember here for the rest of our talk. How we got to the variants of macro H2A2 is that we did an initial kind of screen using clinical data sets. We were looking for histone variant genes whose transcriptions was sufficient to stratify GBM patients in multiple cohorts. Okay, and this is what we got: very small stratification of GBM patients in multiple cohorts. This is not totally unusual for GBM. GBM is a very brutal, incredibly lethal, very quick type of cancer with a 5-year survival rate of 5%. So we never get the nice, or it's very rare to find the nice stratifications of patients we observe in leukemias or other cancer types. But Anna started really looking a little bit deeper into these data sets, and what she found was that if she separates patients based on their membership to the 3 main GBM molecular subtypes, which are classical, mesenchymal, preneural, she finds the macro H2A2 transcriptional levels are very good at stratifying patients in the preneural type of the disease. Okay, so you can see here low levels of macro H2A2 are a negative prognostic factor. Almost no patients make it past 2.5 years, whereas some of the patients with high levels of macro H2A2 can live almost a decade. Okay. Also very interesting here is preneural subtype constitutes about 1/3 of adult GBM cases, and is characterized by PDGFR amplification, which Anna just spoke about in her presentation. And these tumors have a very oligodendroglial progenitor cell-like transcriptional profile. Okay, so they're very interesting tumors, a little bit different in the classical and the mesenchymal. And we try to assess from how good of a predictor macro H2A2 transcriptional levels are to predict prognosis of a patient. So we benchmarked the positive prognostic factor, high transcription levels of macro H2A2 against IDH1 status. Tumors with IDH1 mutations do much better, have much better prognosis than IDH1 wild type tumors. Okay, so this was used by us as a reference point. And then as a negative prognostic factor, we used age. It's very well known that patients over the age of 50 tend to do more poorly than younger patients. Okay, so these are 2 criteria used in the clinic actually when a patient is diagnosed. And as you can see here, macro H2A2 high transcription levels of this gene are actually a better prognostic factor for good prognosis than IDH status. Okay, so it's something that could be of clinical interest down the road. At this point, we have this kind of functional, this kind of association, okay, between macro H2A2 transcriptional levels and survival of patients and prognosis. What we really wondered was if macro H2A2 had an active functional role in determining what we were observing in the clinical cohorts. Okay, so Anna created, she took some of our patient-derived primary cultures from GBMs, and she created these beautiful, stable, DOX-inducible knockdown systems. And we put the cells in orthotopically in the brains of mice. And what we saw was basically this nice phenocopy of what we saw in the clinical data sets. Okay, so low levels of macro H2A2 were negative prognostic factors for this mice. We're at Cancer Stem Cell Lab, so first thing we thought about was, okay, we should probably do some stemness assays to see if macro H2A2 is doing this by modulating stemness. So we did limiting dilution assays in vitro. When we knock down macro H2A2, we see an increase in stemness. okay, higher self-renewal. We then did the opposite experiment where we overexpressed macroH2A2 using a DCAS9 VPR system, a very clean system, and is getting almost 10-fold increase in macroH2A2 expression compared to scrambled guide RNA control. No effects on macroH2A1, the paralogous gene. And what we see is the opposite. We see that stemness goes down when we overexpress macroH2A2. Okay, so this data in vitro suggests the macroH2A2 represses self-renewal and stemness. We did a small study in mice. We did LDAs orthotopically in the brain of mice. As you can see here, at the lowest cell dose of 1,000 cells, there is a stronger tumorigenic potential when we knock down macroH2A2. Okay, so everything is really consistent. Again, laughable cohort. We just worked really hard to inject seven to 10 more mice. I think 7 mice more per cohort. So we should have the final data on this for over the next 3-4 months. These experiments are not very trivial because it's a brain surgery every time for each cell dose. And how does it do this? How does macroH2A2 affect stemness? So we went back to our genomics approaches. Anna did RNA-seq on cells that have knockdown. So these are primary cultures from GBM patients. We knocked down macroH2A2. What we really saw was that well-established stemness signatures published by Suva when he was in Brad Bernstein's lab really show that macroH2A2 downregulation increases the expression of this stemness signature, okay, as would be expected. We also saw positive enrichment for genes associated with a preneural phenotype. We also found a positive enrichment of oligodendrocytic signature genes in the differentially expressed genes. And Anna then started digging a little bit more, and she saw that oligodendroglial progenitor cell markers like PDGFRA or COL20A1 are actually expressed at higher levels when we knock down macroH2A2. So please keep in mind PDGFRA and COL20A1 is going to come up again over the next few slides. Okay. How does the macroH2A2 affect expression of stemness genes? Apparently, it represses, it attacks epinencer elements. So we did attack-seq on primary cultures transfected again with the knockdown for macroH2A2. What we saw is most of the changes in chromatin accessibility occurred in epinencer elements. Some of them got repressed, some of them got gained accessibility. One of the ones that got accessibility was a putative enhancer element in COL20A1, so this OPC marker. Okay, so you can see here, knockdown macroH2A2, accessibility goes up. We kind of tested to see if this was a real enhancer by looking at if there was expression of enhancer RNA from this locus, and we actually found that when we knock down macroH2A2, this intronic region that becomes accessible expresses high levels, much higher levels of this eRNA than the control cell. So it looks like it's the enhancer is actually getting activated. And just to make sure what we were seeing is not an artifact in vitro culture, we did a single cell attack-seq on surgical GBM resections. And we did that for a few patients. And what we found is again, that just a long story short, the accessibility of this putative COL20A1 enhancer only occurs in a subgroup of cells that tend to cluster separately from the others. So it seems like there are some cells that have this enhancer element active, and they're probably more OPC-like, and others that are not. Okay, so it could be irrelevant. It seems to be irrelevant, biologically relevant for GBM, not an in vitro artifact. What does this mean for the patient, right? So that's what the director of our research institute always asks me when I present my data. So it means that macroH2A2 high patients tend to do better than low patients. Why is it? It seems like they're responding better to therapy. So Anna started stratifying patients based on whether they receive no therapy, chemotherapy alone, radiotherapy and chemotherapy, or radiation therapy and chemotherapy. As you can see here, especially for patients that only received radiotherapy, there was a very strong effect of macroH2A2 transcription on survival. Okay, so patients with high macroH2A2 levels seem to be responding better to radiation or concomitant standard of care therapy. Okay, so this kind of suggested that, cells that have high macroH2A2 levels may be more differentiated, less stem-like, seem to be responding to treatment better. Okay, they might. So we did a screen for epigenetic drugs to identify compounds that can increase macroH2A2 levels. Okay, so the reasoning here is if we can increase macroH2A2 levels, we might improve the chances of a patient to respond to therapy. Okay, so We did this work with Doug Mahoney here in Calgary. He's got a beautiful robotic facility for screening. It was all high resolution confocal microscopy screening based on immunofluorescence for macro H2A2. Anna wrote a code to automate the quantification of macro H2A2 levels in each cell, in each well. And what we found were a few drugs that actually increased macro H2A2 levels significantly. And one of them was our old friend MI3, which is an inhibitor of the interactions between menin and MLL1. And we've had a little bit of interest in this drug previously. We published a couple of papers showing that menin inhibition reduces stemness, both using primary cultures, freshly dissociated tumor cells. It seems like it's working. But the mechanism by which it works was not known. Okay, so we wanted to dig a little bit deeper into what menin is doing. First, we really, again, confirmed that menin inhibition increases macro H2A2 levels, okay, at the transcriptional levels, at the protein levels. And then Anna subjected the cells with sublethal concentrations of menin inhibitor. I think it was an IC10 or IC20. And what she found is when she adds menin to the cells, two key PDGF genes that are OPC markers, PDGFR and COL20 and 20A1 become repressed. Okay, so it just feeds back onto this OPC type of network. We validated the downregulation by MI3 at the transcriptional levels, especially for PDGFRA, transcriptional level at the protein level. Okay, so MI3 increases macro H2A2 levels, decreases on OPC stem-like transcriptional network. It's all very consistent. So, you know, we're incredibly interested in pursuing this drug further. A clinical trial using a menin inhibitor called KO539 just started for patients with for some types of blood cancers. So it's something that's not that's actually in the realm of clinical applications potentially, if it's warranted by further preclinical work. And overall, we really found that macro H2A2 can stratify, especially the proneural patients with GBM. And probably that's all because macro H2A2 antagonizes stemness properties in GBM. Okay, so that's kind of my talk. There is more to the story than this, but that's what I wanted to present in 20 minutes or so. I probably went over time, sorry. Just want to really acknowledge the work that Anna did and she's doing in the lab. It's been a fantastic work on so many levels. She was helped by a fantastic undergrad in the lab, Anna Bobin. We did really spectacular over the last couple of years. Francesca Mauro, especially a special shout out to her because she did difficult LDAs in vivo orthotopically. Really like to thank all the collaborators, the Stand Up to Cancer team. It was fantastic to work with them over the last few years, and all of our funding sources. Happy to take questions. Hopefully I didn't overwhelm all of you with all that information.
So thank you, Marco. That was fantastic, and I'm glad that it seems like your cat might have a few things to say as well. I don't know, but
I locked him up in the study here. He's not happy.
But there are a few questions in the chat, so I'll just start with them in the order that they've been presented. Okay. So the first question from Matt Lawrence is: do we know which remodeler, if any, is involved in the deposition or removal of macro H2A?
No, especially in the context of GBM. I am not, to be honest with you, we didn't look into it. So there might be some information out there. Maybe Anna is better, maybe Anna knows.
The only one that's associated with it has been like the FACT complex, and that's about it.
Okay, thank you. So a question from, I'm just going to say Jack. So really interesting work. How does the effect of H2A, macro H2A enhancer accessibility interact with IDH mutations? Of course, we know that IDH mutations via TET2 can impact enhancer accessibility through DNA methylation alteration. Yeah, so do you know if there's any relationship there?
Yeah, so it's actually an interesting thing to bring up, okay? So I think clinically speaking, we observe the effect of macro H2A2 mostly on the preneural type. Most of these cases are probably IDH1 mutant, or lots of them. The cell cultures, the primary cell cultures we were using for our experiments are IDH1 wild type. Okay, so I think overall we find that the effect on microH2A2 seems to have a little bit more of a universal role. The reason why it might be a role that's primed, especially in the preneural subtype, we don't know why that might be, but it's an incredibly interesting question. It would be difficult to assess, actually not that difficult to assess experimentally. You just gave us something to think about. Thank you.
I've also looked at the survival analyses for IDH mutant GBM specifically, and there's no survival benefit of macro in that subpopulation.
So it's a more generalized effect.
Okay, so the next question's was GIB04 your first hit? If so, why not follow this one as a Jumanji C demethylation inhibitor?
Yeah, so there were multiple reasons. So one of them is, personal interest. We've been thinking about menin inhibition for a few years, so we were kind of happy to see that the drug came up. The other reason was when we started searching the literature, we found that again there was a novel clinical grade compound to target menin MLL1 interactions, and so we were like, okay, so then maybe something we can test down the road in our preclinical models in vivo, and then maybe bring it to the attention of our clinical colleagues. So there were multiple reasons for this. I understand that the menin inhibitor was not the first hit, but they were all pretty close, right? So if you go back to that slide, it's not like they were all at a very significant effect of macroH2A2 levels. But yeah, that's definitely a good question, right? So and then some of these PARP inhibitors in GBM had not very good luck in clinical trials. I'm not quite sure what the reason is, if it's in terms of patient selection and from the molecular point of view, or if it's really biological. So we kind of skipped over those ones, and we kind of settled on the menin inhibitor.
Excellent, thank you. So Carolyn Brown asks, have you looked at macro H2A1, and what is the difference between A2A1 and A2A2? Annie
and I were talking about this, whether to include some slides on that. But we saw no effects on macro H2A1 on survival and with transcriptionally speaking. So it seems to be the effects really seem to be residing in the macro H2A2, which is interesting because in melanoma, Emily Bernstein reported that macro H2A1 and to a certain degree also macro H2A2 might play a role. Okay, so there might be some kind of cancer context that makes one variant or the other the important one. In our hands and in GBM, we really saw solid effects, solid associations between macro H2A2 and GBM biology, and not so much with macro H2A1, which made our life also very difficult in terms of reagents. Anna did an impeccable job with the antibodies and antibody validation. And I don't know how many antibodies we went through to detect one, to use, to validate one that was specific to the macro H2A2 variant over macro H2A1. We did a ton of work on that. And it's probably why most of the papers on macro H2A variants, I'll just say variants, it's because it's difficult to have the right reagents to look at each variant specifically. So yeah, it's all about macro H2A2, Carolyn.
Okay, so next question from Steve Billow. So would you say that the treatment regime is selecting for a subpopulation of cells or forcing a chromatin state change? That's a good question.
Yeah, that's a very good question. So I'm just going to talk from what's known about the biology from what's known from the some of the models we've seen, okay, of GBM and other brain cancers. So it seems like, do you guys give me 1 second? I'm just going to let the cat out because.
Oh, come on.
All right, so that's, it's the first one, okay? It's the first meeting, so it's okay to have some interesting things happening. So the question was, so the data on with preclinical models and mouse models really shows that when you add therapy to a mouse, the cells that are more stem-like are left behind, and then they reactivate as soon as the therapy is ended. and they started constituting the tumor after that. Okay, so there was beautiful work from, again, the Dirk's lab on medulloblastoma. They found that in the sonic hedgehog subgroup, SOX2 positive cells are quiescent. They reactivate after therapy, repopulate the tumor. Okay, so they expand massively in numbers, repopulate the tumor. And there is some evidence that might be the case for GBM2. There was work from the Rich lab showing that the cells that are more stem-like, they're just left behind, okay? And they're just basically getting ready to restart the tumor. In terms of patient experience, that seems to be the case too. So there was work from Michael Taylor's lab, for instance, that showed that even from the genetic point of view, basically the key genetic subclones that are found in relapse for some brain tumors, at least, it might not be universal, but for some brain tumors, were clones that were already present at the diagnosis. There might have been a very minor clone that was very detectable from sequencing technologies, which then expanded. Okay. Definitely Peter Dirks also had the Nature paper I mentioned, 2017. It was, the paper was extraordinary because he used single cell viral barcoding of GBM cells, put the cells in mice, and kind of followed the chronal dynamics over time in multiple recipients with therapy. And this seemed to also be the case, support the idea that there was some kind of neutral event based on these stem-like cells that were pre-existing. Okay, so the properties didn't seem to change. It was selection for a pre-existing cell type or phenotype. But at the same time,
Excellent.
I haven't done the experiment, so I don't know if temozolomide might actually changes chromatin architecture. Definitely mutagenizes the cells to an incredible degree, but I don't know about chromatin.
Excellent, thank you. So in the interest of time, I think we'll just do one more question because we're just almost at the top of the hour. So Louis asks, are the effect of MI3 suppressed upon macro H2A knockdown?
Yeah, so we're doing this experiment right now. So and we're actually going in vivo with this. So we're gonna do both the overexpression and the knockdown in vivo. So we need to do those experiments. We can do them in vitro quickly. We literally just talked about doing this a few weeks ago. It took us a while after COVID to kind of restart our cultures. These are all primary cultures, so you know they're slow to restart and stuff. So it's not a great excuse. Everybody's going through it, but it just slowed us down a little bit over the last couple of months. But we can do them easily. Those experiments. Very good question. Thanks.